So my group is the Research Data Analytics and Integration and Analytics group. We are part of a CTSI, a Clinical Translational Science Institute. These are grants given out to academic medical centers from the National Centers for Advancing Translational Science at the NIH. Those CTSI organizations are made up of multiple branches. So there's an educational branch, there's a regulatory branch, there's a community outreach branch, and our group is part of the informatics branch. And we are a service organization for researchers to provide data management and analytical services for their research studies. So I'm going to go over LabKey at URMC, a little bit of philosophy. This is how we implement LabKey in the projects and solutions. And then I'm going to give a generic overview of strategies that we've been using to implement a data management solution in our context. This includes clinical data capture, specimen management, and then integrating experimental data. And finally, what the PIs really want are the reporting and analytics. And I'll touch on future work. So our group supports a variety of projects. These can be multi-center grants, industry-funded research projects, biobanking, departmental or division-wide implementations, and then reagent databases. The areas that we've helped do this in are infectious diseases. This is HIV, AIDS, influenza, RSV, respiratory syncytial virus, autoimmune diseases like rheumatoid arthritis, psoriasis, lupus, lung development. So we have a biobank looking at infant lung development, cancer biobank, clinical interventions in autism, public health, and these are hot topics these days, smoking and vaping, and maternal and children's health. So a variety of research areas and a variety of research types. I will overview our two main systems. We are on a path to, as other people have said up here, of having multiple LabKey instances. But we've been using LabKey since 2010. And our main system, which we call BLISS, the Biolab Informatics System, has over 90 observational or registry studies, 16,000 participants, almost 500,000 samples, and we do integrations for omics and wet bench type assays. I had mentioned our lung biobank, so this is Brindell, and it's going on to a renewal. So we've got the renewal, and so we are building out additional functionalities to be able to share these samples with research communities. So what is our philosophy for implementing LabKey? So as a service organization, we don't just build solutions and walk away. Our goal is to actually participate as active members of research teams. So we are the informatics side of a research. And everything we do is user and process and workflow oriented. So we start from grant submission, all the way through data management planning, all the way through IRB approved protocols, and working on case report form design and REDCap. We also will help with biobanking and getting the processes set up so that you can label your samples correctly. All that data gets integrated with the clinical data. And then finally, enhancing data stewardship and security. So the Federal Information Management Security Act, these have controls on your systems and how they can change and who has access. And then innovating and incorporating best practices. So we are constantly trying to achieve the best tools we can to help our researchers. And you can't have a philosophy slides without actually talking a little bit of philosophy. So this is Conways Law. It says that organizations which design systems are usually constrained to produce designs that are copies of the communication structures of those organizations. This is usually used in the software world to kind of have a pun at software design, but it's really meant to be a sociological observation. And this is true even in science organizations. So the way that science is done is going to be done in a way that is a copy of the structure of the local structure. And so from this, you can take two takeaways for the factors that are driving LabKey adoption. going from a single lab to collaborative science. So a lab that was focused solely on publications for their work now have to start working collaboratively across departments. And single purpose to discovery and reuse. So this could be clinical data that was captured for a specific grant or publication. And now, 5 years later, they actually want to find out where that is and reuse that. Or it's experimental data that they want to go back and discover what's available already and reuse that data. So this is our local research context. So I think Vincent did a good job of talking about the systems that are operational in his environment. And so for research, non-clinical trials, this is kind of our context. And so I'm going to go through each piece of this and talk about how we implement LabKey and the integrations here. So this is, on the top here is mostly the operational system. So the clinical coordinators are doing data entry in REDCap. The labs are actually doing biospecimen management in BSI. And then we have, the outputs of doing the science, right? You have DNA sequencing and you have other kinds of assay data. So in REDCap, I'm going to highlight kind of the technical tools we use that are in LabKey already or that we are going to write ourselves and how we use them. So the first thing is, of course, LabKey provides a really nice REDCap module. This will import data in the CDISC ODM object data model format. It's an XML format. We then have a data integration module, which we write SQL queries and ETL processes. And this allows us to do things like subsets the data, reformat the data, because the way that the clinic wants to enter the data might not be the way you want to analyze the data. So we have an abstraction layer. We can think of the REDCap import as kind of the staging area. And then what we call the final study, the end user facing study. And in the middle are SQL queries and then a linked schema. So that allows you to take schema and data that are available in one container and make those queries available across containers. And then in the final study, we have ETL definitions to actually load into final study data sets. Now, you can imagine this is one study, but you could do this multiple times for multiple studies. And so one example of this is our departmental-wide implementation. So we have clinical studies that are ongoing, doing data entry, and they're interested in specific questions. Now, the chair of the department says, well, I actually want to aggregate all that data, de-identified, and I want to mine it. So I want to do cohort discovery. And not only that, I want to take all samples that have been consented and put them in a central bank that anybody can request in the department to do research. And this layer in between actually allows us to do that, because we can take specific queries for specific purposes and link those across containers and do a centralized repository. In addition to the REDCap provided module in LabKey, we've done some work to develop additional functionalities and interactions with the REDCap system. So we have a Java module where we've implemented a REDCap service, and this allows us to do custom imports and exports of records, download file attachments that are in REDCap, run reports and show those in LabKey, and as well as download study configuration data, so the events, the arms, and the form event mappings. This cuts down on the amount of manual configuration we have to do in that end user study. On top of the API service, we've written custom ETL task definitions. So these are ETL definitions that run as server code inside of a Java module. One requirement we've had is taking assay data and pushing it back into REDCap to share with external collaborators. And so we have an ETL task that will take a query and push that back into REDCap. The second thing we've done, which is a more recent feature, is to actually take files that are attached in REDCap and pull them into LabKey, as what LabKey has a data structure called data classes, and then process those some way inside of LabKey. So here's an example of what the data class might look like. We have information on the record ID, which is the subject ID in REDCap. which arm and which instance of the form this file is downloaded from. And then we have processing data, processing status. If it fails, we have the reason why it failed, and then again, the file link that's been downloaded. We can then take this using the Service API and push back the status into REDCap. So now our clinicians don't have to leave REDCap to enter data and look at the process. So example, a lot of data coming from Excel from our vaping study. They're going to be vaping into an instrument to detect volume and length of puffing. And so instead of making them enter the clinical data in REDCap and then go over to LabKey and drop a file, we can keep them in REDCap and still do all the data integration. On top of REDCap, or actually before we started using REDCap, we've actually developed a electronic data capture framework built on top of LabKey's JavaScript API. So these would insert directly into LabKey data sets and allows us to do more custom things that you can't do in REDCap. So here's a SV scalar vector graphics interactions as well as auto-calculated fields. I'm going to spend a little bit of time on specimen inventory and how that interacts with LabKey. We have a variety of sources of specimen information. Going back to Conways Law, the organizations that collect, or the labs that collect specimen data, are going to do it in a way that their communication patterns, right? So a lot of labs are focused on what they're doing at the bench, and so when we come in to help, we'd have years and years and years of Excel data, and that's their specimen inventory. LDMS is a system that we've migrated away from, and then BSI is the biospecimen inventory system, which is our current biobanking software. This is an operational system for our labs. So this is where they're doing accessioning and processing of samples, freezer and location management, integrations with Bartender. And so Bartender is a software that will do barcoding and label printing. And so what we do is we have a one-way interaction currently with BSI. We pull data nightly and apply custom availability rules. What we really want is a two-way communication between the two systems, like we have with REDCap, such that when specimen requests get created, it will create what's called a requisition in BSI. And so this will allow us to, well, I should preface this, is that it's actually easier for the lab to go into LabKey and find the specimens they want. The user interface is much better. And so we tend to train our labs to do specimen requests to pull those samples. The problem with that is that it doesn't update BSI and mark those samples as unavailable. And so being able to have this two-way interaction between the two is something that we're looking forward to. Now we'll move on to instrument and assay data. So we take instrument-generated data like Bioanalyzer and NanoDrop. We've created custom assay providers so that we can pull in automatically the electropherograms, as well as highlight for the labs those samples that look to be out of range. And so this helps them get out of Excel and to be quickly communicate better what is going on with the samples and where they drop out in the process. So a lot of the times they may send a certain amount of samples to sequencing, but only so many come back because of QC failures. And the question is at what point did those samples drop out? We've also built custom assay providers for managing Illumina sequencing data and runs. So here's an example of an assay where we're just capturing the metadata about that sequencing run. So we know what flow cell, what lane, and what index it came from, as well as the specimen ID. And this actually links back to the BSI records in the study so that we can pull in the primary type, subderivative type, and then a derivative type. So we can actually do data discovery and reuse. So 5 years from now, they can say, well, what CD4 immune cells do we have RNA-seq on? And we have that data. Our goal right now is to use the cloud module to then put this into Amazon Web Services S3 scalable storage. So that's something we're in process and that we currently use. We also host file shares. So these are external reports that are generated by other groups and returned to us as part of the analysis. And so here's an example of what's called a multi-QC report, along with all of the parameters about the computational workflow and general statistics about the quality of the data. And so we can make this available for our statisticians and our lab people to understand the quality of the data before analysis. Now, here are the things that's really the carrots and the hooks for the clinical people. How do we, it's boring, they think, to get all the data in there, right? They already have a process that they think works besides, the barriers to communication. So we tend to build very specific dashboards and visualizations that help them understand the quality of their data, the quantity of their data, and be able to slice and dice that data. So here's an example. This is an industry-funded project looking at RSV incidence. And so we've set up a dashboard very easily to click on a link to go to REDCap to enter the data. Clinical summaries of the incidence of RSV in our population, and then help links. So we actually use tours in LabKey to do pop-up tooltip walkthroughs of our user interfaces. So when a new clinical coordinator comes on, he or she can quickly go through and learn how the UI works, where's the data she's looking for, and et cetera. And then as well as any graphical information that the principal investigator might want to see very quickly. Another example of a dashboard is our lung biobank, Brindell. So we expose the demographics and the available samples that are available for requesting, as well as imaging data processing information. Interactive reports. So one of the advantages of customizing for LabKey is that we can actually begin to tell the clinical coordinators what data is actually missing. And this speeds up the communication loop so that when the statistician gets involved, the data is clean, the data is ready to be analyzed, and there's no waiting. So they can quickly come in here at any time and see the status of what data is available and actually click these icons and go directly back to REDCap. The second one here is a combination of general LabKey plotting along with some custom JavaScript to make it interactive. So this is the bar plot of, again, missing forms across the enrollment visit, and they can click the bars to see the list of subjects that they should go and QC. Real-time reporting. So this is from Brindell again. We can provide overviews of the summary of the sample types and demographics of our subjects. And the last one here is real-time reporting. So this is an example of a biomarker study for detecting viral versus bacterial infections, which is a very important thing if you're going to treat with antibiotics or not. And so we are working with clinicians to adjudicate clinical cases and giving them a summary of the breakdown of what their population looks like before we then send that on for sequencing. And these are the kinds of things that really hook the clinicians to coming back to LabKey and seeing the value. Future work that we have is integrating with additional clinical data sources, so clinical data warehouses, medical record systems, better integrations with Omero. Omero is a slide viewing application. So this is H&E slide staining, multiphoton, fluorescence. And so we run an Omero server, and we had just stood up the web viewer. And so the idea here is that we would generate thumbnails that would be viewable in LabKey. You would click a link, and it would take you right to the slide in Omero. We are starting to do a lot more neuroimaging studies, and so being able to handle the brain imaging data structure format is something that we were interested in. And then integrating with computational workflows, as Vincent said, is something that we would like to do. And so these are Galaxy or Common Workflow Language type analysis external to LabKey. And so for acknowledgements, I'd like to thank everybody in our team. The LabKey folks have been really, really helpful. The larger CTSI Informatics group, as well as our High Performance Computing Center for Integrated Research Computing and all of our funders. Thank you.